Welcome back. Myself, Dr. G. Prasanna, working as an assistant professor in the Department of Genetics and Plant Breeding at School of Agriculture, SR University, Warangal. Today, we'll discuss about the most important topic, that is about the aim, scope, and objectives of the plant breeding. In this lecture, we'll discuss about the what is plant breeding, what is the aim of the plant breeding, and also we'll discuss about the most important topic, that is about the objectives of the plant breeding. Next, coming to the lecture outline, here we'll discuss about the introduction of the plant breeding and next objectives in the plant breeding and significant achievements in the plant breeding in different crops we'll discuss. So first we'll discuss about the definition of the plant breeding. First, the plant breeding is defined as it is the art, science, and technology of improving the genetic makeup of plants in relation to their economic use for the mankind. It is nothing but the plant breeding is nothing but it is a genetic improvement of the crop plants, which is useful for the which is useful for the mankind is nothing but plant breeding. As well as the plant breeding, it is also defined as the art and science which tells us ways and means to change the genetic architecture of the plant so as to attain the particular objective. Or it is also defined as the art science of improving the heredity of plants for the benefit of the mankind. Or it is also defined as it deals with the genetic improvement of the crop plants, also known as the science of the crop improvement. So here we are having the four different definitions for the plant breeding. Next, coming to the aim of the plant breeding. Aim of the plant breeding, the main aim of the plant breeding is to improve the characteristics of the plant so that they become the plants will become more desirable agronomically as well as economically, and generally the specific objectives will be varied depending upon the crops under the consideration. Next, coming to the most important topic, that is about the objectives of the plant breeding. So the most we are having the 14 different objectives of the plant breeding. The first one is the higher yield, and the second one is improved quality, and the third one is abiotic resistance, and the fourth one is abiotic resistance, and the fifth one is change in maturity duration or earliness, and the sixth one is determinate growth, and the seventh one is dormancy, and the eighth one is desirable agronomic characteristics, and the ninth one is elimination of the toxic substances, and the 10th one is non-shattering characteristics, and the 11th one is synchronous maturity, and the 12th one is photo and thermoinsensitivity, and the 13th one is water adaptability, and the 14th is varieties for the new season. So these are the 14 objectives of the plant breeding. We'll discuss one by one. So first breeding objective is the higher yield. So out of all 14 objectives of the plant breeding, the high yield is the most important objective of the plant breeding. Generally, the yield will be in the different forms, whereas it is in the form of grain yield. We can see grain yield in case of rice, wheat. In all different crops, mostly we can see the yield in the form of grain, as well as fodder yield. The fodder yield we can observe in case of bursim and lucerne and bursim crops, whereas fiber yield we can observe in case of cotton crop, whereas the tuber yield we can observe in case of potato and sweet potato. And next is cane yield we can see in case of sugarcane crop, whereas oil yield we can see in all different oil crops like sesame, sunflower, mustard, and a groundnut. Depending upon the crop species, the yield will be in the different forms like grain yield, fodder yield, fiber yield, tuber yield, cane yield, or oil yield. And generally, this higher yield is obtained by high yielding varieties or hybrids. So this is about the high yield. And next is improved quality. Improved quality is nothing but after high yield, the quality is also one of the most important breeding objective. Most important objective in the plant breeding. Generally, the quality characters will be different for each and every crop. So here you can see the example like grain size, color, milling and baking quality, and weight. And here also some of the examples we are having for the quality traits, that is cooking quality in rice. Generally, we will prefer medium amylose type varieties because they are having less stickiness, so that mostly we'll prefer the medium amylose content varieties. And next is malting quality in barley. And next, color and size of the fruits, and nutritive and keeping quality in the vegetables. And next is protein content in the pulses. Generally, the pulses are having the higher protein content, and they are having more amount of lysine, threonine, uh, lysine, threonine, tryptophan. But these pulses are low in sulfur-containing amino acids, that is methionine, cysteine, and cysteine. So, and next is oil content in case of oil seeds, and fiber length, strength, and fineness in cotton crop. So this is about the improved quality. Generally, the quality is also the one of the second most important objective in the plant breeding. This quality is also differs from each and every crop. Here are the some of the examples for this. And next, coming to the abiotic resistance. Abiotic resistance is nothing but generally the crops will suffer from some of the, generally there are two types of the stresses of two types, that is abiotic stress and biotic stress. Generally the crops will suffer from both abiotic and biotic stresses. Because of this, the plant because of this biotic and abiotic factors, the plant will result in the will result in yield losses. So to avoid that, we have to develop the varieties which are having more resistance to both abiotic stress as well as the biotic stress. So here the first is abiotic resistance. Generally, we have to, generally the plants will suffer from these abiotic factors like a drought, soil salinity, extreme temperatures, heat, wind, cold and frost. So the breeder has to develop the varieties which are suitable for for this abiotic factors, for this environmental conditions. That is about the abiotic stress. And next is biotic stress. Biotic stress also, here also the plants are suffered by some of the pest and diseases, because the plant was attacked by this different pest and diseases. It will automatically it will result in the yield losses. So the genetic resistance is the cheapest and the best method of minimizing such yield losses. And the resistant varieties are developed through the use of resistant donor parents which are available in the gene pool. This is about the biotic resistance and abiotic resistance. And next is change in maturity, duration, or earliness. Generally, earliness is nothing but the crop will mature earlier, because if the crop will mature earlier, it also escapes from the different abiotic and abiotic factors also. So this earliness is also one of the most desirable characters which are having several advantages. It requires if the crop matures earlier, it requires less crop management period and less insecticidal sprays and permit new crop rotations, and it also often extends the crop area. So generally, because of this earliness, the development of the wheat varieties which are suitable for the lean planting has permitted the rice wheat rotation. And generally, if because of the changes in the maturity duration, here we can see the examples for this. The maturity has been reduced from 270 days to 170 days in case of cotton crop, and from 270 days to 120 days in peach and pea, and from 360 to 360 days to 270 days in case of sugarcane. So this is about the change in maturity duration and earliness, which are having the several advantages with this. And next is determinate growth. Determinate growth is it is nothing but the development of the varieties with determinate growth is most desirable in some of the crops like mung bean, peach and pea, and cotton crop. This is about the determinate growth. And next is dormancy. Generally, at the time of maturity, if the rains will occur, the seeds will germinate even before the harvesting in the standing crop. That is nothing but dormancy. So here we can observe the dormancy in case of some of the crops like green gram, black gram, barley, and pea. So this period of dormancy has been introduced in these crops to check loss due to the germination, and in some other cases, however, this may be desirable to remove the dormancy. So this is observed in some of the crops like green gram, black gram, barley, and pea. So this is about the dormancy. And next is desirable agronomic characters. Desirable agronomic, if we'll select the if we'll select the varieties with desirable agronomic characters, automatically the yield and everything will be more. So we have to While developing the variety, we have to take care about this agronomic characteristics like plant height, branching, tillering capacity, growth habit, erecta trailing habit. So these are the some of the agronomic characters which are often desirable. And for this, we can see the examples. Generally, the dwarfness in cereals like rice and wheat is generally associated with the lodging resistance, and they are having the better fertilizer responsiveness. And next is the tallness and height tillering and profuse branching are the desirable in case of fodder crops. In fodder crops, the plant should be tall and it should have more number of the tillers and more branching, so that these characters are more desirable in fodder crops. So this characters also, this desirable characters also changes according to the each and every crop. So this is about the desirable agronomic characteristics. And next is elimination of the toxic substances. Generally, some of the crops are having all the desirable characters, but because of the presence of some of the toxic substances, it is making unfit for the human consumption. So elimination of these toxic substances from this varieties will make them safe for the human consumption. So here you can see the examples of the some of the toxic substances which are present in different crops. Like here you can see the neurotoxin in kesari, that is Lathyrus sativus, scientific name. It leads to the paralysis of the lower limbs, and whereas erythric acid from brassica, which is harmful for the human health, and gossypol, it is also toxic component from the cotton seed. So, this removal of the neurotoxin from the kesari and erythric acid from the brassica and gossypol from the cotton seed, which will make them fit for the human consumption. So, removal of this toxic substances will increase the nutritive value of these crops also. So, this is about the elimination of the toxic substances. And next is non-shattering characteristics. We have to develop the varieties which are having resistance to the shattering in case of green gram crops, because this shattering of the pods is always a serious problem in green gram. Next is synchronous maturity. Synchronous maturity is nothing but it is a maturity of cross species at one time. Generally, in case of green gram, copy and cotton, we cannot observe the synchronous maturity. Here we can go for harvesting of the crop for 4 to 5 times. So to avoid that, we have to develop the varieties which are having the synchronous maturity in case of green gram, copy and cotton crop. And next is photo and thermo insensitivity. If we develop the varieties which are having photo and thermo insensitivity, which will helps to grow the crops in different countries, that is nothing but photo and thermo insensitivity. So here, because of the development of photo and thermo insensitive varieties of wheat and rice, they have permitted their cultivation in new areas also. Now the rice is also cultivated in the Punjab, whereas wheat is also a major rabi crop in the West Bengal. And next is wider adaptability. Adaptability is nothing but it is the suitability of the variety for general cultivation over a wide range of environmental conditions is nothing but adaptability. This adaptability helps. It is also a major objective in the plant breeding, which will help in stabilizing the crop production over the regions as well as the seasons. And next is varieties for the new season. Generally, maize is grown as a kharif crop, but now we are growing because scientists are now able to grow this maize as both rabi crop as well as a summer crop. And similarly, the mung bean is also now grown as a summer crop in addition to the main kharif crops. So these are the 14 most important breeding objectives of the plant breeding. And next we'll discuss about the major achievements of the plant breeding. First we'll discuss about the semi-dwarf wheat. Here the semi-dwarf wheat was developed by the scientist Norman E. Borlaug. Norman E. Borlaug, he is the father of green evolution in the world. He has introduced the semi-dwarf wheat varieties from the CIMET, that is Mexico, and these semi-dwarf wheat are having the dwarfing genes from the Norin 10, that is Japanese variety. And next is Indian varieties are Kalyan Sona and Sonalika, and here they are having the rust resistance and higher yield. This is about the semi-dwarf wheat. And next is semi-dwarf rice. Semi-dwarf rice is derived from the Adizio Ozen, that is a Japanese variety from Taiwan. And so here the TN1 and the IR8, which are introduced in India in 1966. And here we are having the Indian varieties, that is Jaya and Ratna. Here, this Jaya is the miracle rice. This is about the semi-dwarf rice. And next is mobilization of sugarcane. Here we are having the two different types of canes, Indian canes and the noble Indian cane and noble cane. This Indian cane is having low sugar content, and this it is also hardy, but noble canes are having the high sugar content. And the major eminent sugarcane breeder is Venkatraman. And the major breeding center for sugarcane is located in Coimbatore, that is the Sugarcane Breeding Institute. And next is hybrid millets. Generally, the ECRI program, that is All India Coordinated Research Improvement Project on the maize, was started in 1957. And because of this, we can see the yield advantage up to 40%. And whereas in sorghum, we are having the CHS series, we are having the CHS 1, 2, 3, 4 for Kharif season, and 5 and 6 for the Rabi season. So we are having the varieties exclusive for both Kharif season as well as for the Rabi season. And next is pearl millet. We are having the hybrid Bajra and Punjab hybrid series in case of pearl millet crop. And next is hybrid cotton. This hybrid cotton, the major eminent scientist for the hybrid cotton is the C.T. Patel. The first hybrid, that is H4, which was released in 1970. In this cotton having both intra and interspecific hybrids, and the exploitation of heterosis is important case of cotton crop for development of hybrids. And next is oilseed improvement. Generally, the soybean is a major oilseed crop. Uh, as we know, the soybean is it is a miracle crop which is having the both which is having high protein content, around the 40 to 45% of the protein content and 20% of the oil content. This is the soybean is also major oil seed crop. And next is Pusa Jai Kisan, which was developed as a somatic hybrid mustard. And next is PM30, which is having the low erucic acid mustard. Next is National Mission on Oilseeds and Palm Oil was started. Next is molecular breeding. Molecular breeding is nothing but here we will be having mark resisted selection. Here we are in molecular breeding, we are having the mark resisted selection, bacterial leaf blight resistant rice varieties, Swarna sub-1 submergence tolerance, quality protein maize, and genome sequencing achievements. First is mark resisted selection. Mark resisted selection is nothing but it is an indirect selection for the it is an indirect selection for the traits. Here the DNA markers are linked to the desirable trait, so that we can identify the trait, and those traits which can be useful for the which can be useful for the further breeding programs. And next is BLB resistant rice varieties, that is bacterial leaf blight resistant lines. Generally, this BLB resistance was obtained by using the that is marker resistant backcross breeding. Here we'll go for the foreground selection, background selection, and recombinant selection. First we'll cross first we'll cross the donor parent and recurrent parent, and we'll get the hybrid, that is F1. Then this F1 is backcrossed with to the recurrent parent up to BC. We'll go for BC1 to BC6 generations to obtain the whole genome of the recurrent parent. And in case of molecular breeding, we will go for the three selections, that is foreground selection, background selection, and recombinant selection. We are having different markers for this all these three types of selections, and then they have developed some of the bacterial leaf blight resistant genes, that is XA1, XA5. These genes are having the bacterial leaf blight resistant genes. And next is Swarna sub-1. This is also developed by using the mark resistant backcross breeding. This Swarna sub-1 is the submergence tolerance in case of rice. It will be the it will the plant the if the plant is having this Varna sub1 gene, they can if they are present in the submergence conditions, they can survive up to 15 to 20 days. So that is about the Swarna sub1. And next is quality protein maize. Quality protein maize is nothing but this is nothing but QPM. This is the QPM is nothing but it is a maize with a high lysine and tryptophan content. Generally, the normal maize is having low lysine and tryptophan content. So, to reduce that, they have developed some of the quality protein maize with a higher amount of lysine and tryptophan content. That is the quality protein maize. We are having the here we are having higher amount of lysine and tryptophan content, and the gene which was responsible for this is OPEC2 gene. And next is genome sequencing achievements. We are having the first generation sequencing, second generation, and third generation, and the new generation sequencing methods. So this is about the molecular breeding. And next, coming to the transgenic crops. The first transgenic crop developed was the tobacco, and next is BT cotton and BT maize. BT cotton was developed. BT is nothing but it is the Bacillus thuringiensis, which was developed by the Berliner. This BT cotton was developed by using developed against the bollworm, the American bollworm as well as the pink bollworm. Because of this BT cotton, the yield of the cotton is gradually increased in case of cotton. And next is BT maize. This BT maize was developed against the maize shoot borer. So these are the two transgenic crops. And next is golden rice, that is vitamin A enrichment. This golden rice is nothing but they have this vitamin A is a um, precursor for beta carotene. So this golden rice is having higher amount of the vitamin A content to avoid the color blindness in developing children. And next is quality protein maize. Here also they have by using the transgenic technology they have developed quality protein maize. So these are the some of the transgenic crops. That is the first transgenic crop is tobacco, and next is BT cotton and BT maize they have developed, and next is golden rice. They have developed with a higher amount of vitamin A content, and next is quality protein maize. And next, coming to the conclusion, here as we know, the plant breeding is the most important discipline in the agriculture that helps to improve the crop yield, quality, and adaptability to meet the growing food and nutritional demands of the population. Though through this systematic steps like creation of genetic variation, selection, evaluation, multiplication, and distribution, the superior crop varieties are developed for the different environmental conditions. And achievements like we are having different types of achievement. We are having so many achievements in the plant breeding like semi-dwarf rice, wheat, and millets we are having, and some of the hybrids and improved oil seeds and transgenic crops. Because of this, they have significantly enhanced the agricultural productivity. And apart from the conventional methods, with the advancement of this molecular breeding and biotechnology, the plant breeding has become more precise and efficient. Overall, this plant breeding continues to be the foundation of the sustainable agriculture and global food security. So again, we'll revise the lecture. That is first, we have discussed about the definition of the plant breeding. Definition of the plant breeding. It is the plant breeding is nothing but it is the genetic improvement of the crop plants for the economic use of the mankind is nothing but plant breeding. And here we are having the different definitions for this plant breeding. And next is same. The plant breeding main aim is to improve the characters of the plant so that whatever the plant we have developed, it will become more desirable agronomically as well as economically. And here we can see the all the 14 important objectives of the plant breeding. That is first is a high yield. It is the most important objective of the plant breeding. Generally, and we can at this high yield by using the high yielding varieties as well as hybrids. And next is quality. After yield, the quality is one of the most important breeding objective. We can see different types of quality parameters in different crops. This like cooking quality in rice, malting in barley, and protein content in the pulses and oil content in the oil seeds, and fiber strength, length, and fiberness in cotton. So this is about the improved quality and biotic and abiotic resistance. Um, Generally, the crop will suffers from both biotic factors, that is, pest and diseases, and abiotic factors like drought, salinity, and extreme temperatures, and freezing conditions, and frost. So, to avoid that, we have to develop the varieties which are having resistance with for both biotic and abiotic resistance, which are available in the which are present in the resistant donors, which are already available in the gene pool. And next is the maturity duration. Generally, the earliness is one of the most important desirable character, because if the plant will mature earlier, they can escape from the different types of biotic and abiotic factors. So earliness is also one of the most important desirable character. And determinate growth. This is also determinate growth is also most desirable in some of the crops like moonbean, pigeon pea, and cotton, because in these crops we can see indeterminate growth habit. And next is dormancy. Dormancy is nothing but if the during maturity time, if the rains are occurred, we can see the seed germination before the harvesting in the standing crop. So we can see this dormancy in some of the crops like green gram, black gram, barley, and pea. And next is desirable agronomic characteristics. We have to select the characters which are having more desirable nature, so that it will helps in improving the yield as well as the quality in different crops. And next is elimination of the toxic substances, because of the presence of the toxic substances, this the nutritive value of those crops will be reduced, and it is also not safe for the human consumption. So we have to remove the toxic substances from crops which are having the toxic components like neurotoxin in Kesari, and erucic acid from the brassica, and gossypol from the cottonseed. And non-shattering characteristics. Generally, the shattering is a major problem in case of green gram. So we have to develop the varieties which are having more resistance to this shattering. And next is synchronous maturity. Here also we have to develop the varieties which are having the maturity of a crop at one time in case of green gram, cowpea, and cotton. And next is photo and thermo insensitivity. If we develop the varieties with the photo and thermo insensitivity, we can grow those varieties in all over the world. So this thing was developed in case of wheat and rice. And next is wider adaptability. Wider adaptability also, if you develop the variety for general contribution for wide range of environmental conditions, it will stabilize the crop production for regions and seasons. And next is varieties for new seasons. Generally, the maize is a kharif crop, but with the help of the scientists, we can grow this maize crop as a rabi crop as well as a summer crop. And the similar thing was happening in the moonbeam, which is grown as a summer crop in addition to the main crop. And here we have discussed that is major achievements of the plant breeding. Here you can see the major achievements in the plant breeding, that is semi-dwarf wheat, semi-dwarf rice, and mobilization of the sugarcane. Here the semi-dwarf wheat, which was developed by the Norman E. Borlaug, that he is the father of green revolution in the world. He was developed the semi-dwarf wheat from Cimet, that is Mexico, and the dwarfing genes are from Norin 10, and they are having the rust resistance and higher yield. And the semi-dwarf rice, it is also derived from the DCO Usin, that is Taiwan, and they have developed TN1, TN1 and IR8, which were introduced in India in 1966, and we are having the Indian semi-dwarf rice varieties, that is Jay and Ratna. And next is nobilization of the sugarcane. Here we are having the two canes, that is Indian cane and noble cane. Indian cane it is having a low sugar content, whereas noble cane is having the high sugar content. And the breeding center for the sugarcane is Sugarcane Breeding Institute, that is Coimbatore. And next is hybrid millets. ECRIP was started on the maize crop in 1957, and it is having the yield advantage up to 40%. And in Sorghum, we are having the hybrids exclusively both for Kharif season as well as for the Rabi seasons. And for pearl millet also, we are having the hybrids, that is hybrid Bajra and Punjab, and PHP, that is which is released from the Punjab, that is Punjab hybrid Bajra, and the hybrid cotton. We have developed the first hybrid, that is H4, which was released in 1970. And in cotton we are having both intra and interspecific hybrids. And the exploitation of heterosis is important in case of cotton crop, whereas oilseed improvement in case of soybean, it is as we know, it is a major oilseed crop and is a miracle crop which is having high amount of protein content around 40 to 45%, and oil content of 20 to 25, around 20% of oil content. And we can observe the somatic hybrid mustard, that is Pusa Jai Kisan. And we are having the low erucic acid mustard, that is PM30. And here you can see the molecular breeding. So here you can see about molecular breeding, and these are the transgenic crops, and this is about the conclusions. Thank you.